Hello everyone. Today we will be solving Cambridge IGCSE Biology Paper 4, October-November 2041. Question number 1. Water is an essential molecule for life. Complete the statements. Water moves into and out of the cells by osmosis. Water is known as because it can dissolve solute. So water is known as a solvent because it can dissolve solute. A leaf was put into a solution. The water potential of the solution was lower than the water potential of the contents of the cell. Figure 1.1 is a stretch of the cell after 3 hours in the solution. So guys, the solution is concentrated. And we know that concentrated solution have low water potential. So definitely water will move out of the cell by osmosis and the cell will shrink. And that's why we can see the cell is shrink. The leaf was transferred into pure water. Sketch the expected appearance of the cell after it had been in the pure water for 3 hours. Draw one arrow on your sketch to show the direction of water movement. So the question says, draw one arrow on the sketch to show the direction of water movement. So this is the direction of water movement. We have drawn the cell target because water entered this particular cell and the water is pushing in the cell wall. Question number C. A plant was not watered for 1 week. Figure 1.2 shows a series of photographs of the plant during the week. Day 0, day 3, and day 7, we can see the plant has wilted. Explain how the lack of water has affected the support of the leaves of the plant shown in figure 1.2. Use the term turgor pressure in your answer. So guys, when question like this says that you have to use a particular term, it is, we have to use that particular term directly in our answer. So the plant, we can see the plant is wilting. Due to the lack of turgor pressure. By the end of the week. Since the cell membrane is no longer pushing against the cell wall. The cells are no longer turgid. Mesophyll cells are not providing support and the cell collapses. the cells the cells becomes the cells becomes flaccid or plasmolized due to the lack of water. Question number 2. Pathogens cause disease. Figure 2.1 shows some cells that are part of human immune system. These cells are responding to one type of pathogen. So we can see Q is basically representing the pathogen. So Q is basically the pathogen. And the molecule T is representing antibodies. S is thereby a lymphocyte. We can also see R has a lobed nucleus and it is engulfing the bacteria, so thereby must be a phagocyte. So both S and R, they are white blood cells. Explain how the immune system responds to an invasion of pathogen. Use the letters in figure 2.1 in your answer. Pathogens are recognized as foreign due to their unique antigen in the cell surface membrane. In the cell surface membrane. In the diagram, S and R are white blood cells. S is a lymphocyte that makes antibodies. T are the antibodies made by S. So we can see T are the antibody that is made by S. Antibody T is complementary to the shape of antigen. Antibody T is complementary to the shape of the antigen. Antibody T binds with antigen on pathogen Q, and this action marks the pathogen. Q. Phagocyte R contains digestive enzymes to digest pathogens. Question number B. A vaccine was introduced in 1942 for a particular disease. Figure 2.2 shows the effect of introducing the vaccine on the number of cases of the disease in one country. Number of cases of disease per million people from 1920s to 1980s. We can see that the vaccine initially has a upward trend from 1920s up until 19 number of cases of disease per million people. We can see from 1920s the number of cases increases, decreases, and then it increases, decreases. In this trend it goes until 1937 or 38. But from 1940 to when the vaccine was introduced, from there the rate of the number of cases of the particular disease kept on decreasing until 1980s, where we can say that from 1974 to 1980s the cases almost became zero. In 1946, the government of the country concluded that the vaccine was successful. Discuss the evidence shown in figure 2.2 for and against this conclusion. So guys, there is a general decrease in number of cases. Per million people from 1942 after vaccination. The number of cases did not return to pre-vaccine level. The number of cases did not return to pre-vaccine level. There are no cases from 1974. We can see from 1974 onwards, there are 0 cases. So all of these points are for. Now we are going to write down the against. Number of cases during the first 2 years after the vaccine was introduced. The number of cases increased during the first 2 years after the vaccine was introduced. The vaccine program took 32 years before no cases of disease. Finally, there are small peaks in cases even after the introduction of vaccine. Guys, moving on to the next question. All living organisms excrete waste products. Figure 3.1 is a photomicrograph of a Naegleria fowleri, a single-cell protoctis that live in watery environments. A is labeled cell membrane. State a feature of N. fowleri visible in figure 3.1 that distinguishes it from prokaryotes. Guys, we can visibly see the nucleus. Prokaryotes don't have a nucleus. State the name and function of structure A. So structure A is cell membrane. And it controls what enters in and leaves the cell. Enters and leaves the cell. Suggest how N. fowleri excretes carbon dioxide. N. fowleri is a single cell organism, so definitely should excrete carbon dioxide by diffusion. So N. fowleri excretes carbon dioxide by diffusion through cell membrane. Urea is a toxin that is secreted by the kidneys in humans. Describe how and where in the body urea is formed. Guys, urea forms in the liver. Due to the breakdown of excess amino acid. By deamination. Deamination is the removal of nitrogen containing part of amino acid in the liver. Guys, moving on to the next question. Question number C. Figure 3.2 shows part of the human excretory system and associated blood vessels. The arrow indicates the direction of blood flow. So D, since the blood is going out through here, so it must be renal vein. E, since the blood is coming through here, so must be renal artery. F, looks like the urine comes out of this particular tube, so thereby it is ureter. G, the urine is stored here, so this is the bladder. H, urine is going to pass out from here, so it's the urethra. Now the question says draw a label line and the letter X on figure 3.2 to show the location of the cortex in one of the kidneys. So the cortex is actually the outer region. So label line and X drawn. Table 3.1 contains statements about the label structures in figure 3.2. Complete the table by stating the name of the structures, identifying the letters that label that structure. Organ that stores urine. Bladder. Bladder is G. Tube that carries urine out of the kidney. Ureter. Ureter is F. Blood vessel with the lowest concentration of urea. Blood vessel with the lowest concentration of urea should be renal vein, because all the urea has been lost in the kidney, has been taken out in the kidney. So renal vein is D. Blood vessel with the lowest concentration of carbon dioxide. Oh, that's going to be renal artery, because arteries have higher concentration of oxygen and lower concentration of carbon dioxide. So renal artery, E. Tube that carries urine out of the body. So that is urethra. And that is H. D. Doctors wanted to investigate the effect of exercise on the excretion of salt. They collected urine from people before and after running a long distance on a hot day. The results of their investigation are shown in table 3.2. Average volume of urine / cm cube before running 1156, after running 569. Average concentration of sodium in urine millimole per dm cube before running 85.6, after running 78.2. Suggest why there is a difference in the volume of urine produced before running compared with after running. Use the information in table 3.2 in your answer. So guys, it's very simple. Before running the race, obviously the person was properly hydrated. But during the race, the person got dehydrated because of all of the urine, because of all of the sweat that came out of the body, right? So we are going to start the answer in this way. So people lost more water as sweat while running, which explains why they have a smaller volume of urine. And at the same time, since they were running, they were not drinking enough water. So people did not drink enough water while running. Finally, more water is reabsorbed. More water is reabsorbed from glomerular filtrate to maintain homeostasis. Which resulted in concentrated urine and smaller volume of urine. Calculate the percentage decrease in the average sodium concentration after running compared with before running. So after running compared with before running, give your answer to significant figures space for working, okay? So the concentration after running, before running is 85.6 and after running is 78.2. So if we want to find out the percentage, we have to compare it with before running. So 85.6 into 100%. The answer comes to be 9%. Describe how the kidney tubules enable the excretion of salt. So salts from the blood move from the blood into the tubule as blood flows through glomerulus. Salt ultrafilters, since it's a very small particle, it can just simply ultrafilter itself through gaps in capillary. Some salt are reabsorbed by active transport. But excess salt remains in the urine. Excess salts remains in the urine. Large plasma proteins are usually prevented from entering the urine. State the name of one protein found in blood plasma. Okay. So one protein found in blood plasma is fibrinogen, because it's the protein that helps coagulation. Guys, moving on to the next question. Figure 4.1 shows a bee with pollen on its legs. Bees are insects that pollinate some flowering plants. They are attracted to the flowers by their color, scent, and nectar. Describe other ways in which flowers and pollen grains are adapted for insect pollination. So for insect pollination, the flower must attract the insect. So there are key features in the flower that actually allows it to attract the insect. So first, we would say insect pollinated flower have large, obvious petals. with honey guidelines. So basically, this is how, the insect is directed towards the honey. And because of the honey, the flower, the insect will enter the flower. The anther or the stigma, the anther or the stigma will be located inside the flower. Any one of them will be located inside the flower because plant wants to prevent self-fertilization. The filaments are stronger so that they are not broken by the insect's arrival. Pollinators must touch anthers to reach nectar. Insect pollinator flowers have sticky stigma to catch the pollen. Sticky stigma to catch the pollen. Next, they also produce large pollen and the pollens are sticky. State whether pollen is produced in a flower. State where pollen is produced in a flower. So pollen is produced in the anther. State the name of the process that produces haploid pollen nuclei. So meiosis produces haploid pollen nuclei. Meiosis is also known as reduction division. So actually, this is also a correct answer. Explain why it is important that the pollen nuclei are haploid. So that diploid number can be restored after fertilization. All right, moving on. Describe how the pollen that is carried by an insect to another flower result in the formation of a plant embryo. Guys, after the pollen is transferred to stigma, it forms pollen tube. The pollen tube grows down the style. The pollen tube enters ovule. The male nucleus then fuses with the ovule and the fertilization occurs. This leads to the formation of zygote. The zygote divides by mitosis to form an embryo. The fertilized ovule becomes the seed. Guys, next question. Describe the advantages of cross-pollination. So the advantages of cross-pollination is that it allows genetic diversity. Cross-pollination allows genetic diversity. Plants are more likely to survive environmental changes, example, global warming. Plants are likely to receive more resistance to disease by cross-pollination. And increase in fitness. And increase in fitness in general. So they will be more able to survive the changing weather pattern and everything. Some people are concerned that genetically modified plants might cross-pollinate with wild varieties of plants. Suggest how farmers could prevent cross-pollination between genetically modified plants and wild varieties of plants. Guys, farmer can grow GM crops in glass houses. GM crops in glass houses. There is one solution, a rather expensive solution. A more cheaper solution will be farmers can use sterile GM crops. So they will not be able to fertilize the wild varieties. State two advantages of genetically modified crops. The advantages, well, they provide additional nutrients. GM crops may provide additional nutrients like vitamin A. And GM crops, some GM crops have this amazing ability. They have resistance to insects, so they require less pesticide. So requires less pesticides. Guys, next question. Milk is a source of some of the nutrients that are part of a balanced diet. Calcium and protein are two nutrients found in milk. Describe the importance of calcium and protein in the diet. So guys, calcium is needed for formation of bones. Bones. Calcium prevents rickets. Calcium is also needed for the formation of teeth. Protein. Protein is needed to repair tissues. Protein allows growth. And also used for making muscles, antibodies, digestive enzymes, etc. Lactose found in cow's milk. Some people do not have the enzyme to digest lactose. State the name of two organs associated with alimentary canal that can produce enzyme. So name of any two organs that can produce enzyme in the alimentary canal. So first of all, salivary glands. They make saliva. Then the next thing that comes in my mind is stomach. Releases pepsin for the digestion of protein. We can write two more, pancreas and small intestine, both of which are additional points. All right, both of which also releases enzyme. Figure 5.1 shows a flow diagram for the production of lactose-free milk. Step 1, collects cow's milk from farm. Okay, step 2, heat milk to a high temperature. So obviously, if you're heating the milk to a high temperature, it destroys any pathogen in the milk. Allows the milk to cool at 20 degrees Celsius. Pour milk into a container of beads containing enzyme. Collect lactose-free milk. Store lactose-free milk in 2 degrees Celsius in sterile bottles. Okay, explain how heating the milk in step 2 in figure 5.1 ensure the hygienic preparation of lactose-free milk. Oh, so basically heating the milk kills all the pathogens. So that's how. So we're going to write down heat kills bacteria and other microorganism. Explain why the milk must be cooled in step 3 before it makes contact with enzymes. You see, heat will denature the enzyme. So heat will denature enzyme. So lactose will not be broken down. We already got our mark. So some additional point will be heat changes the shape of the active site. So the enzyme will not feed the substrate. So the enzyme will not feed the substrate. State the name of the enzyme used to make lactose-free milk in step 4. So to make lactose-free milk, we require lactase. Moving on. Suggest why the enzymes are kept in beads in step 4 rather than mixed as an enzyme solution with the milk. So if the enzymes are in beads, they can be reused. So enzymes in beads can be reused. and it's cheaper that way. Milk is produced by mammals. Explain the advantages of newborn mammals of breast milk. So guys, breast milk contains antibodies and provides protection against pathogens. Breast milk is easy to digest, does not contain any additives as well. Breast milk is sterile and delivered at correct temperature directly from the mother. Breast milk does not require any preparation and it is free. Breast milk also increases the bonding with mother. So that's one of the advantages that needs to be considered. Guys, moving to the next question. Explain why breastfeeding mothers are advised to drink plenty of water and avoid excessive alcohol consumption. So basically, we must understand water is needed to make the milk, needed to produce breast milk, because 90% of it is water. So alcohol, when drank in excess, obviously can pass to the baby in breast milk. Alcohol can delay the development of the baby. So the brain development will be hampered. Okay, guys, moving on to the next question. Sensitivity is one of the characteristics of all living organisms. Define the term sensitivity. Now, in our last few question papers, we have done this answer several times. Sensitivity is the ability, sensitivity is the ability to detect stimuli in the internal or the external environment. To make appropriate response. To make appropriate responses. The eye is an example of a sense organ. Define the term sense organ. So sense organ are basically a group of receptor cells responding to a specific stimuli. Adrenaline is a hormone that is released in fight or flight situations. It causes a change in the eye. Complete the table 6.1 by stating the part of the eye that change when adrenaline is released into the blood. So action, muscle that relaxes, part of the eye. So muscle that will, so basically we know adrenaline will cause dilation of the pupil. So if the pupil needs to dilate, the circular muscle of iris needs to relax. So circular muscles of iris, muscle that contracts to dilate the pupil, radial muscle of iris must relax, and this will cause the pupil to widen. Okay, guys, so that's the end of the video. Hope you have learned something new. And we will be uploading this kind of videos, and we'll keep on uploading this kind of videos as we carry on. If you have any question paper that you have recommendation that you want us to solve, then mention it in the comment, and subscribe to the channel and to support us.